CEO and founder of Thrive Bioscience in the Boston area. And Marty, we started this company because we found an incredibly ignored segment of life sciences, which has almost no data, almost no software, is done almost completely manually, and yet is surrounded by other parts of life sciences that have all these tools, and that is cell culture. So live cell biology is based on cell culture. It's incredibly important. It's the basis for most research. The cell is incredibly important. And yet Uber knows more about me and us than we know about ourselves. And it's entirely predictable that this area is going to be brought into the 21st century with tools just like are available in almost all other fields. It's just that it's been ignored. So we have two products on the market. Next slide, please. We have two products on the market, there are 19 instruments, and more than half of them we've sold to pharma and biotech. And we basically sell to researchers almost entirely for drug discovery. And they buy our instruments because we give them data that they never had before. We give them documentation almost automatically. And so when experiments don't work, they can go back and look at why. A higher percent of their experiments work, and they're able to make new insights and have data sets that have never been available before. And so the applications are for assays, for CRISPR, which is cell engineering, for CAR-T, for growing tissues, which is the fastest growing area of cell culture. So we've had sales of about $2 million, and in the next 12 months, we expect $6 million in sales, and onward in 5 years to over $150 million. So we think we have the team to do it. My own background, I've started 11 companies, 4 of which have been multi-billion-dollar exits. So I've started companies. My first 3 were in AI, in software. I started a company called HNC Software, which spun out a public company called Retech, which was acquired by Oracle for $1 billion and is still a major part of their AI efforts. And then Fair Isaac acquired the rest of HNC Software. And there we were basically pooling data from our customers and giving them better answers with better algorithms. We're doing the same at Thrive. Then I started, after HNC, a machine vision company where we applied it to cytology labs. And we did the same thing for cytology labs that we're going to do with Thrive for cell culture labs. Cytology labs read pap smears. For 65 years, things were done almost the exact same way, just like in cell culture. And we formed Cytic, and our instruments are now being used around the world. 90% of all pap smears in the US and most countries use our instruments. We went public and the company was acquired for $6 billion. And we're going to do that here. We have a lot of the same people, a lot of the same investors. So we have really significant collaborations. One is with a very large Japanese pharmaceutical company to develop an advanced tissue imaging system. And we have strong collaborations with Harvard Stem Cell Institute, the Broad Institute, Stanford Stem Cell Core, University of Texas Medical Branch. And these are key opinion leaders. And we're building basically now software modules that automate key workflows within life sciences. And we're applicable to almost all parts of life sciences. Because these instruments have not been developed before, and there's been so little innovation in cell culture and live cell imaging, we've amassed a portfolio with 78 patent applications, and 25 have issued. So basically, scientists are going to have to buy our products, or somebody's going to have to buy us. So we've raised $29 million to date, mostly from family offices, the largest of which is the Estee Lauder family. We have several angel groups, such as Life Science Angels in California. And we're currently finishing up a $5 million convertible note that will convert into the Series B, which we expect in Q3. And we'd love to talk to people who could help us fill that last $1 million. We're using those proceeds for sales and marketing. We're just launching our second product in the family. And we've signed a letter of intent to acquire a small profitable company that we work with actually a lot as one of our vendors. We'd love to finish up that financing quickly. So the problem is that there's really a lack of imaging, of data, of documentation, analytics in this incredibly important area, which is drug discovery, which is bringing medicines to people. And there's almost no data collection. It's occasional, it's insufficient. The data and images are not comparable. They're different in each lab, and they vary tremendously. So researchers spend several hours a day looking through a microscope, looking at millions of cells, hundreds of thousands of stem cell colonies. And guess what? They put the plate back and no data is retained. Occasionally they'll snap some photos using a digital microscope, but it's a tiny portion of all of the cells that they're growing. They're not capturing things in a rigorous basis. And so there's a lack of control, there's a lack of tools, and this is what is leading to the reproducibility crisis. One of our customers said that chairman of department at University of Texas Medical Branch, leader in infectious disease research and COVID-19 research. He talked about what the problem is with current tools, which is they are failing us because they hurt the cells, only give us 2D single point in time images, and they don't capture contextual data or enough data. Next slide. So this has led to what's known as the reproducibility crisis. And Amgen did a study, had up to 100 scientists, and issued the bombshell results, which is of the 53 landmark studies that they tried to duplicate. They could only duplicate the data in 6 of them. So 90% of these studies were not science. If you can't duplicate the data, it's not a fact. There's been study after study. The best that has been shown is that half of the studies are duplicatable. Next slide, please. So our solution is instruments and software that provide automated imaging, analytics, documentation, so we have reproducible cell culture, stem cell culture, and tissue culture. That same person who talked about the current tools and what they're lacking said after using our instrument, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissues in 3D over time with remarkable images. And another researcher said, for the first time, we have high quality, consistent data to vastly improve experiments and enable machine learning, data mining, and breakthrough insights. Next slide, please. It really is all about the data, and that data is just not available in live cell biology. And so the needs are for extensive and sufficient data. You need very large data sets for AI, for machine learning, and they need to be in a form that can be read by people and also by computers. And we need comprehensive data. Right now, the data that's collected on live cells is biased. If something looks interesting, a picture is taken, rather than of all the cells in all the wells over time. So what we produce is comparable data every time we scan a plate, and we take tens of thousands of images. And we do it in multiple dimensions because biology is not flat, it's 3D. So we use 60 focal planes or more, 2 to 50 microns apart, so that we can capture the whole organoid, or the most important part of what's going on in cell culture. And we don't hurt the cells. Next slide, please. So Marty raised a very important point, which is binary decisions by the FDA are put many companies at risk, and we don't have that. So we're selling tools to all of the researchers who are developing drugs that will go through the FDA. We ourselves are not regulated. And so it's a picks and shovel kind of situation. So this is our product line. On the left is our benchtop instrument. It's about $100,000, sits on top of the benchtop, and in minutes takes 20 to 90 gigabytes in 10, 15 minutes. For the first time, tens of thousands of images are available and made easy. They're very accessible. The product that we're just launching now, the Cell Assist 50, combines the same imager with a 50-plate incubator. So around the clock, automatically using robotics, it's imaging plates. So you have these incredible time series of images, and because we get back to the same place, you're actually able to follow the biology. We're trying to be like the biology. Most of our focus now is on the software modules, since the hardware is done in key areas such as viral plaque assays, machine learning toolkit, tracking single cells. Next slide, please. So for the first time in live cell biology, we're producing enough data and the right data for machine learning and breakthrough insights. And kind of our magic sauce is the imaging and the fact that we are capturing rapidly very high resolution and in-focus images, which is actually a problem in the industry. And we're capturing all of these stacks of images so that we can see up and down. And we can do something that has been a holy grail that nobody else has been able to do, which is we can find and track a single cell over time. So again, you can follow the biology. And this is incredibly important after cell engineering, where you have a single cell or single cell sorting. And we give you comparable images. We take the same set of images every time that we see that plate, and really all plates. And so for the first time, we have genomics quantities of data. Our customers are generating 2 to 3 terabytes a week of data. Next slide, please. Their competition is mostly digital microscopes, and they lack the large quantities of reproducible data that we're able to provide. They are not able to track a single cell. They don't have the advanced focus algorithms that we do, and don't fit into the workflow the way that we do. Next slide, please. So we have installed base of 19. Most of them, about half of them are in the US, and about half are in Asia. And the industry breakout is about 60% are in industry, which is contract research organizations and pharma. By field, cancer and immunology is the largest use of our instrument, followed by neurology. And the most common activity is drug screening. So studying the effects of a drug on a tissue or cells and comparing hundreds of drugs, we're able to automate all that and collect all of that data. And about a quarter of our customers are using it for production of cells. And our customers are evenly split using it for cells, stem cells, and tissues. Next slide, please. So the power of data is, I mentioned one of the problems is the lack of data, and that has resulted in there being incredible mislabeling rates. Next slide, please. Oh, thank you.
Is this right?
Yep, this is it. Sorry. And up till now, so the rates have been established by the FDA, NIH. It's being 14% to 20% mislabeled cell lines. So that means somebody thinks they're studying prostate cells, but they're actually studying liver cells, which is part of the reason for why we have such low success rates in drug discovery. But as you can see in this chart, just by having data and being able to see how your cells compare to all the others, which has not been available, you can see in this case that there were outliers. This was from a major research institute in the United States, and it turned out that those were in fact mislabeled cells. Most people don't think it's a problem they have, but everybody has. Next slide, please. So this is an example of use of the instrument for viral plaque assays, which has been used in COVID-19 research, and the department chair who said Thrive will change the world, it's because of these images. And so for the first time, without staining cells, we're able to see the viral plaque that forms. So basically an overlay of viruses are put onto a lawn of cells, and we count the holes. And we've had to stain and touch the cells again in order to do that. And we haven't been able to see anything before or after. And so for the first time, they can actually characterize these holes and how they form. And there's been tremendous data in them. Next slide, please. So this is an example of what's called a scratch assay. So literally a scratch is made on a plate of cells. And we currently watch how they fill in, how the wound is healed. But the problem is you have to do it manually. You have to estimate what the volume is. You can't track and compare many combinations of drugs. So this is pretty astounding. There are like over 15 combinations of drugs that were used on these scratches, and we automatically calculated the rate and the amount of healing that occurred. Doing it manually, there's never been really the ability to accurately count or have this kind of throughput to compare 15 drugs at a time. Next slide, please. So this next slide is images of a single cell being tracked over time, which is something that every biologist has wanted to do. And you can basically see the cell doubling until it becomes a colony. And this is incredibly important for well-known and important technologies like CRISPR, where you engineer a cell and it's a single cell and then you have to grow it. And right now, without our instrument, you can't find it. You don't even know if it's alive for a couple of weeks.
We got about 7 minutes.
Okay, next slide, please. So this is organoid images, and this is the fastest growing area of cell culture because rather than doing drug discovery on a plate of cells, we're now putting those cells together into tissues or organoids. And this gives us more information than just these single cells on the bottom of a plate. It's between those and animal models. So we now have a better model where we can put together, say, a tumor model where we have the 5 to 10 cells that are actually found in those tumors and screen drugs on that. And we are the best instrument for imaging organoids because we have these stacks of images. Next slide, please. So the cell culture market is a huge market, $29 billion per year. The vast majority of it is pharma and biotech. And we're selling through direct distribution into this market. And the drivers for us is that our product's applicable to almost all areas of life sciences. And our objective is really to become the premier source of images and data for all researchers and to develop a database that we can monetize. And we're expanding into tissue culture. We didn't know that we could do that back a year or so ago. So we're expanding into additional areas. And the big change for us will be the production of cells. And we're beginning to do that. I mentioned a quarter of our customers are doing that. The bioprocessing market is even bigger than the research market. And we're one of the few tools that can provide data. Next slide, please. Yep. So I mentioned my own background. It's my co-founder is much more impressive. Alan is responsible for most of the sequencers. There's a huge company, multi-billion-dollar company called Illumina, and he developed those sequencers. And basically, for 25 years, he brought to genomics huge amounts of reproducible data, and that's what we're doing for live cell biology, which has been ignored. Next slide, please. So I mentioned we've raised $29 million to date, and we're currently in a convertible note. We've had nice increases in pre-money and post-money. And we'd like to wrap up this convertible note in the next couple of weeks. Next slide, please. In terms of what our exit's going to be, almost all life science tools companies end up being bought by Thermo or PerkinElmer or Sartorius, Agilent, Danaher. We've been contacted by the majority of the 15 likely acquirers. And the multiples are really phenomenal, 35 times 12-month trailing averages for public research tools companies, and 10 to 15 multiples of last year's sales in M&A. So we'll go either way. Next slide, please. So I just wanted to end with a few customer quotes. Remarkable resolution. We see things we have never been able to see before. Tracking single cells and colonies in 3D is game-changing for drug discovery. Everything is documented. We no longer guess at who did what and lose valuable time and money. This is what cell culture should look like. It's all about the data, and this field, like all fields, needs to have databases and software and tools. And it's not very complicated. We're just really bringing some automation and best in practice tools to this ignored area. So thank you very much. Love to take questions if we have time ready.
We have about 3 minutes. I have, is there a question?
Yeah. Excellent. Really interesting. What for an institution to acquire both the hardware or the software, let's say they want the whole package, if that's the right term, what's the cost, and would be an annual maintenance cost for the software?
So we sell two instruments. One's $100,000. One's about $250,000. It comes with all of the software that allows for all of these different workflows. We're going to change that. We are, at this point, we want to get lots of use cases and key opinion leaders. But one of our investors was the founder of Viva Systems, the largest clinical trial software company, entirely subscription. So we're going to move our software to a subscription, which will mean that we could sell the hardware, which will still sell as capital equipment, but at a much lower price point, under $50,000 instead of the $100,000. And since our main focus is software, 8 out of 9 of our engineers are software engineers. We're going to be generating a lot of modules that we're going to sell independently.
So your software will run with other machines?
No, it only runs on ours for now, but that's a really important point, Marty. We do plan to be on every microscope someday.
Right, because that would make sense if you sell a software as a service product, and then you can just vacuum up data that other people won't.
Exactly, we want
more precision.
Exactly, we want the data from all those microscopes. It would not be as functional by far as ours, because they wouldn't have all these stacks and wouldn't be able to follow a single cell. But we still do a lot with our software that's not done. And so we want to really be the aggregator of all this data.
And does your existing equipment use kind of like fuzzy data, AI, to kind of get the more refined imaging? Because
Yes.
I mean, imaging is always a part that's data science to it, right?
So we have a really incredible AI group, machine learning and image processing people, and probably have focused more on this field and getting sharp images than anyone else. So our autofocus images are really incredible. Those images of a virus, no one else has been able to take those, because they usually end up focusing on debris, which we don't do. So it's an important part of what we do. We don't tend to advertise it, but it is an important part. There was a question.
Yep.
Can you talk a little bit about competition?
Sure. So the main competition are digital microscopes, which everybody has competition, and we certainly do, which is really manually done, collects very small amounts of data. There are a few scanners that are mostly for fluorescence, as opposed to, we don't touch the cells, we don't add anything to them. Those scanners, such as IncuSight, have a single focal plane, and so they really can't image tissues or suspension cells. We're the only instrument that can image cells in suspension, which is important for immunology and CAR-T. Each of our images are 5 megapixels instead of 1. We take about 100 times more images. We do it about 20 times faster. And because we focus on the software, it's easy to use. The important point here is biologists, and probably the reason this has been ignored, is biologists are not good engineers or instrument people. And so you have to make it really easy for them. And so we're really marrying 2 disciplines, good software and imaging.
One follow-up question regarding the data. Do you have data rights to the data? We own it? We shared with the researchers?
So they own their data, and we have a license to use the data internally on a blinded basis. Our vision is to produce the largest database in cell biology. And so we've tested whether they would allow us to build a database of sharing images. And so far, all of our customers, except one, so it's 18, are willing to do it. So we're testing that, and we'll make a part of our standard T's and C's that we not only have rights to data, but as long as it's up to the point where they add something proprietary, so just while it's in the cell culture stage, that we'll be able to put those on a blinded, no names basis on a database.